Bismillahirrahmanirrahim. Assalamu alaikum. In my previous lecture, we discussed the basics of immunity, types of immune response, and an overview of lymphoid and myeloid cells. Today, we are going to start with the role of cellular components immunity, significance of lymphocytes, and the differences between innate and adaptive immunity. Starting with the antigen-presenting cells. These are the heterogeneous group of immune cells that mediate the immune response by presenting and processing antigens for recognition by certain specific cells, for example, macrophages, dendritic cells, and B cells. They gobble up pathogens and present antigens. These cells also release cytokines, which are the tiny molecules that attract other immune cells to the site of infection. Among antigen-presenting cells, macrophages are the large cells with a round to oval nuclei. Next one are the dendritic cells. These are the professional antigen-presenting cells, which are located in the skin, mucosa, and lymphoid tissue. And they are also the key regulator of immune system. The B cells are also known as the B lymphocytes, and they are a type of the white blood cells. They play part in humoral immunity, which is a component of the adaptive immune response or the adaptive immune system by secreting antibodies. Macrophages play a fundamental role in phagocytosis, antigen presentation, and cytokine production. Phagocytosis uses the extension of the cytoplasmic membrane to surround the particle or the microbe and enclose it in a membrane. Thus, this enclosed microbe is killed by the reactive oxygen species and lysosomal enzymes, for example, proteases, nucleases, etc. Macrophages also act as antigen-presenting cell. So how does it do it? The macrophage takes up the antigen by the process of phagocytosis. Antigen is broken down into small fragments. Then a class 2 MHC molecule, it binds to these fragments. Then this class 2 MHC molecule along with this antigenic fragments, they are expressed on the surface of the cell. And these cells, MHC class 2 protein along with this antigen, they interact with the T helper cells. The macrophages also produces cytokines, which are the specialized chemicals that attract other immune cells to the site of infection. Now the question arises how the macrophages get activated. For phagocytosis, antigen presentation, cytokine production, macrophages has to be activated. These are initially activated by the bacterial lipopolysaccharides, bacterial peptidoglycan, and bacterial DNA. These substances, they interact with the toll-like receptors present on the macrophage cell surface, and they signal the cell to produce cytokines. The macrophages, they are also activated by gamma interferon, which are produced by the T helper cells. Gamma interferon, they increases the synthesis of MHC class 2 molecule, and thus they increases the microbicidal activity of macrophages. The monocyte, they only circulate in the blood, but whenever they are fixed to the tissues, they become macrophages. And so they are named according to the site where they are found. The alveolar macrophages, they will be seen in the lungs, osteoclast in the bones, histiocyte in the lymph nodes, and the microglia in the brain. Dendritic cells are also the antigen-presenting cells, and they also express the class 2 MHC molecule, just like macrophages. And they present antigen to the CD4 T helper cells. These cells have many long, narrow protoplasmic processes which help them in efficiently engaging the foreign material. After capturing the antigen, they migrate to the local lymph nodes where they present antigen to the CD4 helper T cells. This diagram shows the function of the dendritic cells. After engulfing antigen, for example, bacterium, these foreign bodies are broken down into small fragments. These fragments are then displayed on the surface of the cell through MHC class 2 molecule. These dendritic cells with the MHC class 2 molecules then migrate to the nearby lymph nodes where they will interact with the CD4 helper T cells. These helper T cells will then be activated and they will start to produce more cytokines in order to attract the other cell to the area of infection to destroy the infected cells. If the MHC class 1 is activated, then this will interact with the CD4 cytotoxic T cell, T lymphocyte, and thus the infected cell will be killed at once. Remember one thing, it all depends on the type of antigen that which class of the MHC molecule, either class 1 or class 2, will be activated. Neutrophils are also the phagocyte, and they have a characteristic multi-lobe nuclear and granular cytoplasm. These cells express a special membrane protein, CD66. Cytoplasm of the neutrophils, they contain two types of the granule, primary and secondary. The primary granules can include the azerophilic granules, which is a characteristic of the young neutrophils. They contain cationic proteins, lysozyme, defensins, elastase, and myeloperoxidase. The myeloperoxidase, it is abundantly expressed in neutrophilic granules, and they play a major role in antimicrobial activity. The secondary granules, they are specific for the mature neutrophils, and they contain lysozyme, NADPH oxidase components, lactoferrin, and B12 binding protein. Lactoferrin has the pro-inflammatory effect, and thus they enhances, thereby enhancing the neutrophilic adhesion. So what's in the granules? This diagram shows that a bacterium or a microbe is ingested by the process of phagocytosis. A phagosome is formed, and phagolysosome is, and the next phagolysosome is formed. So here you can see the lysosome contains the digestive enzymes which digest the bacterial cell wall, and in addition to the antimicrobial proteins. So this figure shows the two-edged sword role of neutrophils in addition to the phagocytosis. At point 5, you can see the cell digests the ingested material. The microbial antigens can thus also be presented on the cell surface receptors, and thus they will enhance the inflammatory response. So this shows the neutrophils can also play the positive as well as negative role at the same time. Positive role is the phagocytosis, and thus the phagocytosing the microbe. The negative role is to trigger the inflammatory response and thus damaging the body's own tissues. Lysozyme employ multiple mechanisms for killing and degrading ingested microbes. These include oxygen independent and oxygen dependent mechanism. Oxygen dependent mechanism is further subdivided into myeloperoxidase dependent and myeloperoxidase independent mechanism. In oxygen dependent mechanism, activated phagocytes synthesize the lysosomal enzymes and various hydrolytic enzymes whose degradative activities do not require oxygen to kill the microbe. In oxygen-dependent mechanism, respiratory burst occurs, which means that oxygen consumption increases, and as a result, reactive oxygen species, superoxide, anions, hyperoxide ions, they are produced, which are microbicidal. This class of the non-specific killer cell include eosinophils, basophils, mast cells, and natural killer cells. Eosinophils, as compared to the neutrophils, they are much less common. This granulocyte has a characteristic bilobe nuclei, as shown in this figure, and the cytoplasm of this cell contains the granules, which stain with the acid dye eosin, and thus it appears pinkish red. Eosinophils are also phagocytic, and they are best known for fighting the large and unwieldy parasites, because eosinophils are much larger than neutrophils, and they have receptors that are specific for the parasites. They also, their granules also contain some major basic proteins, and this is a potent toxin for helminths. Unlike neutrophils and eosinophils, basophils are the non-phagocytic. They stain blue with dihematoxylin, And like eosinophils, they can be helpful at combating large parasites, but they can also cause inflammation in asthma and allergic response. And finally, the mast cells. These cells are also known phagocytic cells, and they are involved in the asthma and allergic responses. Natural killer cells, they are a part of the innate immune response, and they are also known as the large granular lymphocytes. These natural killer cells, these are identified by the certain surface proteins, which are the CD56 and CD16. Natural killer cells are the large lymphocytes which, with granules, which are activated by the IL-2, interleukin 2, and interferon gamma. These cells, they target the infected cells and the intracellular organism by releasing cytotoxic granules in their cytoplasm directly into the target cell through perforins and granenzymes. Now, how does the killer kills the infected cells? So here you can see the natural killer cell. It attacks the enemy cell or the target cell, releasing the perforins, which made a punch hole in the target cells. And after punch hole, the granenzymes are released, and these are taken into the target cell. The enemy cell dies by the process of apoptosis, and this apoptotic cell is then engulfed by the macrophage. So now the specific mechanism of the immune system, which is the third line of defense, this includes the lymphocytes and antibodies. Up till now, we have discussed the cells of myeloid progenitor cell. Now let's discuss the other group, that is the lymphoid progenitor cells. These cells, they migrate to thymus, where they develop into the T cells. B cells are also developed in the bone marrow, and then both B and T cells are released into the blood, lymph, and the lymphoid tissue. Lymphocytes, they arise from the lymphoid progenitor cells, and these cells are part of the adaptive immune response. The T lymphocytes, they are subdivided into T helper cell 1 and T helper cell 2. And this, they are also known as the CD4 T helper cells and the cytotoxic T cells. So this slide shows a schematic diagram of how the T lymphocyte differentiate into Thi1, T helper 1, and the T helper 2 cells, which then leads to activation of the macrophages and the cytotoxic T lymphocytes or the B cell activation to produce the antibodies. This is a complicated process which requires a series of proteins molecules to activate the cell sequentially. This table shows some of the major markers for identification of the T helper, T cytotoxic, and B cells. For example, CD3 is positive in T helper and cytotoxic T cell, but negative in B cell. And the CD40 is positive in B cell, but negative in both the cytotoxic and CD4 helper T cells. In this slide, there is a brief summary of development of the B and T lymphocytes. In bone marrow, cells are differentiated into pre-B and the pre-T cells. B cells get matured within the bone marrow. These mature B cells are then released, are taken into the lymph nodes via lymph circulation. On the other hand, the pre-T cells, they are taken into the thymus for further maturation via lymph or via lymph circulation, and from there the thymus, the pre-T cell, here they mature into the or they develop into a mature T cells, and from thymus the mature T cells are taken into the lymph nodes via circulation. Now, how does the T helper cells, which are a class of the T cells, they differentiate into subclasses of the T1, T2, and T17 classes? This differentiation is based on the cytokine profiles, which are elaborated by the specific cells. For example, we'll see here, the T1 releases IL-2 and interferon gamma, which leads to activation of the macrophages. And the T17 here, it is activated by the IL-6, which is released by the naive T cell. And the Th2 helper 2 cell, it is activated, which leads to release of the cytokines IL-4, 5, 6, and 10. And these then leads to activation of the B cell, which then produces antibody. This is the table of comparison between subclasses of T cells, that is the B cell, C cytotoxic T cells, and the T helper cells. All the cells respond to pathogens via their specific surface receptors. The B cells, they do not require they do not require MHC molecules to present the antigen, but they do it directly. Whereas the the cytotoxic T cell, it requires MHC class 1, and T helper cells, they require the MHC class 2 molecules for antigenic presentation. The co-stimulation or the second signal is produced in order to strengthen the interaction of the MHC and other molecules for antigenic presentation. Cell-mediated immunity, which is a part of the both immune responses, are specifically effective against fungi, parasites, and certain cancers. These immune cells also play their role in rejection of the organ transplants. For example, delayed or the type 4 hypersensitivity in tuberculosis. This is due to their cell-mediated immune response.